Started. This is Barb's PhD dissertation defense. And I just wanted to say a quick word about how we're going to handle the question and answer section. It's going to be a little different today. When Barb has completed her seminar, we're just going to pause for about a minute. And we're going to do that so that people who would like to ask a question in ASL can raise their hands. And when they raise their hands, I will promote them to panelists so that they have video access. And the reason we're going to pause for a minute is because that just takes a little bit of time. So if you get promoted to panelist, you'll have a tiny bit of downtime where the meeting will kick you out for a second and then readmit you. And so don't worry when that happens. And then for everybody else who does not need to ask their questions in ASL, please go ahead and use the question and answer box at the bottom of your screen. And then Barb will be going through those and moderating her own Q&A section. So I'm going to put all these instructions in the chat as well. So thank you all for being here. And I'm going to turn this over to Bruce.
Thanks, Teresa. I am going to, in turn, hand it over to Jenna Sullivan-Stack and Brittany Parson, who want to go through a brief summary of Barbara's time here at OSU. So go ahead, you guys.
Well, welcome everyone. Welcome. Before you get to sit back and learn about science with Barbara, we really wanted to take the time to share about Barbara's journey throughout this PhD program. Barbara is incredibly adventurous. She loves to play games, and when she plays, she always wins. But she's also sweet as candy. And we decided to kind of depict and portray her journey using this classic board game, Candyland here. So as you can see here on the Candyland board, Barbara is bringing a friend along on her adventure. This is typical Barbara behavior. She's an incredible friend. She's patient, she's funny, she's fun to talk to, and incredibly generous with her time and expertise. And so many people in our lab, in Corvallis, and all over the world are lucky, so extremely lucky to count Barbara as a friend.
Barbara, during her PhD, has shown us just how fearless she is. During her PhD, she not only acted in a play, she went into the scariest haunted house you've ever been in, was totally calm. She's helped her friends over difficult terrain and even dodged the waves to put tiny little electronics installed into the intertidal. Fearless.
Throughout Barbara's PhD journey, she has just demonstrated the open-minded nature and open-hearted nature that she shares with everyone. She's always up for going new places, trying new things, whether it's Oregon, like those pictures on the top, or Iceland or New Zealand, or traveling underwater. She's been on some incredible adventures, and she is always in style. Her style is always on point. Well, almost always.
As you will soon see, Barbara has pioneered new ways of studying kelp in our lab. She's a statistical wizard. She has that rare quality of being able to explain her very complicated science to others in a way that they can understand. Just to give you a little taste of her expertise, In the last 5 years, she has given more than 60 presentations.
And when Barbara joined the Mingy Lab, the way we communicated really changed. It improved. So it changed for the better.
You know, we used to always be talking over the top of each other, and a lot of our communication would break down. Barbara has really shifted the way we look at communication. She's made us all better listeners, always stressing the importance of waiting for people to finish talking and sharing our ideas clearly.
And she made it. She made it. And now we all get to sit back and see the culmination of these 6 years. Congratulations, Barbara. And Barbara says, thanks you guys.
Okay, so I'm going to be brief. Barbara has a lot she wants to tell us today. Barbara came to us from Northeastern University, where she got a master's with a former postdoc in our lab, Tarek Gouillet. He's now an associate professor there. And she had done her undergraduate work at the Rochester Institute of Technology. She arrived here immediately after doing her defending her master's in 2014, and pretty much jumped in feet first right away. To me, the remarkable thing that Barbara has done, in addition to doing a really great PhD, she's got 4 chapters, all of which are eminently publishable. She has, at the same time, done what some might think would be another full-time job, in that she has been involved in bringing science to the deaf communities. So she's formed her own, she's got her own website, Atomic Hands, which is aimed at bringing science to people in the deaf community. And she's also works with an organization in Massachusetts that has the goal, well, it has several goals, but one of which is to help develop scientific language in ASL and to bring science, especially to young folks in high school as well as into college. To me, that's just mind-boggling that she's been able to do that and do everything that she is about to tell us. We've loved having Barbara in the lab. We're really going to miss her. I was hoping she'd still be in this area, but she just informed me she's moving back east fairly soon. And in conjunction with that, she'll be moving on to a postdoctoral position with Jen Cassell at the University of California at Santa Barbara, working remotely on a marine reserves project. So without any further ado, Barbara, take it away.
Awesome. Thank you, everybody. Thank you, Brittany and Jenna and Bruce, for the really warm and heart-touching and inspirational introduction. It makes me look back on all of my time here and really just know how much I'm going to miss you all. So thank you. I'm going to go ahead and share my screen. Okay. Okay, to start off my talk today, I want to open with a metaphor. And the metaphor will kind of frame my talk, and I'll come back to it from time to time throughout my talk. So throughout our lives, we are constantly taking in information about our environment. And one of the ways that we can record and take in information about our surroundings is using cameras. Of course, cameras have become even more common now than ever. All of our smartphones have one in them. And so most of us have now become pretty familiar with the basics of photography. And you know how to use different lenses to capture what you want to capture. So you see a beautiful landscape and maybe you want to take a closer look at it. You can use your zoom to zoom in and get a more detailed look. If you zoom in again, you'll get even different details, even if it's the same landscape that you're looking at. The same can be said for time-lapse photography. So you're looking at a landscape in the case of time-lapse photography, taking a picture of the same exact area, but at different time points, thus getting information about how that particular landscape is changing through time. So we'll use that photography metaphor throughout my talk as I take a closer look through various lenses to look at what's going on in the rocky intertidal zone with marine macroalgae. We'll be looking at and exploring the environmental factors that influence macroalgae and the responses of those macroalgae as we zoom in and take snapshots at different spatial and temporal scales. So to give you just a little overview of how my talk will go today, I'll first, just mention that we do have 2 interpreters here. They are providing the voice interpretation into English for those of you who don't know American Sign Language. And you don't see them on the screen, but I am connected to them through a separate Zoom. So if you see me looking off screen down below here every once in a while, you'll know why. It's because I'm just communicating with our interpreters here. And we also have captioning available if you prefer. Just click that little button, CC button at the bottom of your screen if you'd like to see the captions. So I'll start by giving you today a general overview of my dissertation, what led to it, and then I'll be talking about 3 of my chapters, giving you an overview of the methods, results, and then some discussion about the results in each chapter. And you can see the little icons here that will guide you as we're going along through the talk. They'll highlight on the progress bar at the bottom, so you'll know which part of the talk we're in and where I'm at in each chapter. And then I'll end by summarizing my findings and giving us a sense of where they might go, implications for future studies. Within any ecosystem, organisms within that ecosystem, just like humans, as an organism in our environment, we're observers. Organisms are observers of their environment. There are various factors, both biotic and abiotic, as well as an organism's life history that will alter the organism's experience, so to speak, of that environment and ultimately alter their responses to that environment. So like I said, there are all these different factors that can alter an organism's experience of their environment. And all of these factors operate on different scales. So zooming into the smallest, lowest level of the organism itself, that would be within the organism itself as an individual, and then moving up to the local scale, which would be anything happening in the environment up to like tens of kilometers. Then moving up to the meso and macro scales, which is whatever's going on in the environment at the scale of hundreds and thousands of kilometers. Oh, excuse me, okay. So for my focus, we'll be looking at marine macroalgae as my organism of study. Marine macroalgae are a diverse group of organisms critical to the productivity, structure, and function of coastal ecosystems. We call them ecosystem engineers because they create habitat, providing organisms with a source of food and shelter. Marine macroalgae also oxygenate water as well as recycle nutrients. They trap and stabilize sediments and form the basis of the world's primary fishing grounds. All of those popular places that people like to go to fish, there's lots of algae there too. Furthermore, they form an integral part of the world's aquaculture industry. So zooming into that smallest, lowest scale, on an organismal scale, factors operating would include demographic traits of the individual, as well as life history stages of the species. Marine macroalgae go through different life stages as they age. They start off as little gametophytes, growing into juvenile sporophytes, and finally growing into adult sporophytes. Of course, marine macroalgae are continually interacting with their environment, such that environmental factors will then be what drives the formation of upper and lower threshold values of tolerance for macroalgae. Macroalgae have tolerance boundaries and thus continuously have to calculate their energy budget, optimizing trade-offs in energy allocation among the demographic traits of growth, reproduction, and survival throughout their life history. And these threshold factors are critical to the species and vary with life history stages. Some examples of factors operating now on a local scale, if we zoom out one, would include biotic interactions such as herbivory, competition, and facilitation. We'll start with herbivory. Communities of marine organisms, of course, include not only macroalgae communities, but also critters. Benthic grazers are some of the most important animal communities within macroalgal ecology. Examples of benthic grazers include limpets, chitons, urchins. All of these play an important role in determining what macroalgal assemblages will look like. If grazing intensifies, for example, beyond a certain threshold, herbivores can then reduce population density as well as decrease algal community diversity. Algae are always competing. They're competing with each other for space and for light. They're competing with other individuals within the same algal species, as well as competing with individuals from other macroalgal species. And they are also competing with sessile animals. Now for that's competition. Now for facilitation, a macroalgal species can, in some cases, enhance the survival of other macroalgal species by providing structural complexity, something other than bare rock, for those other species to recruit and then grow on. The mix of these interactions can also change as the individual changes due to age as well as environmental history. If we zoom out one more scale, factors operating on a mesoscale include, for example, coastal upwelling. Here along the Oregon coast, coastal upwelling is one of the main oceanographic processes that shape coastal macroalgal communities. So what's happening when you have coastal upwelling? Well, basically, you've got alongshore winds that are moving water around, creating upwelling favorable conditions. So that warmer water that's on the surface that is depleted of nutrients will move away from the shore, offshore, and in its place will be welled up colder water that is replete with nutrients. And within these upwelling regimes, because of that cold water bringing up lots of nutrients from depth, it tends to produce areas of high biological productivity and will also cause fluctuations in temperature, nutrients, and light, which in turn influence the strength and direction of species interactions. If we zoom out one more level, factors operating on a macro scale would include, for example, the El Nino Southern Oscillation. also known as ENSO, the El Nino Southern Oscillation, is one of the major climate patterns that governs the dynamics of macroalgae populations. The ENSO cycle causes variability in temperature and precipitation, which can have cascading effects then on coastal communities. ENSO has two phases that alternate every 3 to 7 years. Looking at the figure here, you'll see the red spikes on the top of the graph representing the warm El Nino phase. That's when sea surface temperatures rise to above average. And the blue phase, the blue La Nina phase, represented on the bottom part of the graph, representing the times when sea surface temperatures go below average. Any ecological process, of course, will have an effect on one scale, but on a different scale, those very effects may look very different. So the strength of ecological processes can be said to be scale dependent. So any given ecological process, the strength of its effects on the environment will vary across time and space. Thus, our ability to precisely quantify and understand the effects of a given ecological process on biodiversity really critically depends upon our understanding of the scales over which they operate. The environmental factors that are important to macroalgae communities and associated with climate change, coastal upwelling, and ENSO are predicted to intensify in the future. We know that macroalgae do have an indispensable role in our environment, yet so far they've really been understudied. Most macroalgae related publications that are out there focus on subtidal kelps. Focusing on subtidal kelps has really left out all the species residing in the rocky intertidal, which have not been studied much, and they should be. Intertidal macroalgae deserve more attention simply considering that their habitats are ecotonal, meaning they have both marine and terrestrial influences, both of which are changing due to climate change. Given our time constraints today, I selected just a few of my chapters to focus on, and I'll share my findings from 3 of my chapters, focusing on 3 common rocky intertidal kelp species in Oregon: Hedophyllum sessile on the left there, Egregia menzicii in the center, and on the right, Postelsia palmiformis, the little palm tree guy. So this is my overarching research question. We know that environmental processes from local to regional levels, as well as species identity, do influence the responses of intertidal kelp communities along the Oregon coast. But specifically how, in what ways do they interact with each other to influence kelp species responses in various ways, and how do those influences change across organizational scales? We'll start with the smallest organizational scale and work our way up. And we'll do an interpreter switch.
So with chapter 2 here, we start at that smallest of scales, the organismal scale, where survival of early life history stages is critical to the successful establishment of future benthic populations. So light availability and herbivory are 2 factors likely to influence the successful establishment of kelps. But the interactive effects of both of these factors on macroalgae, especially in the context of coastal upwelling, has not been well studied. So in this chapter, I ask whether the effects of herbivory and light over the population dynamics of Hedophyllum sessile can be strengthened by coastal upwelling. In upwelling ecosystems, nutrient pulses from coastal upwelling indirectly underlie competition between phytoplankton in the water column and the benthic macroalgae beneath it. So upwelled nutrients stimulate those phytoplankton blooms, increasing the turbidity of the water and shading intertidal substrata. This shading may create a recruitment bottleneck for annual cohorts of juvenile kelps because the light availability is decreased for them. And because only those surviving juveniles will pass through the bottleneck and grow into adults. Nutrient availability is also often correlated with grazing intensity. So high nutrient levels can stimulate the growth of benthic macroalgae, which through bottom-up effects can increase grazing intensity. And in areas with high upwelling and frequent phytoplankton blooms, herbivores graze on juveniles, which also creates a recruitment bottleneck. So then we have a combination, right, of shading and grazing that may reduce juvenile survivorship and ultimately end up reducing adult densities overall in the population. So this was a project I ran last year, May through November of 2019. Here I studied a single kelp species, Heterophyllum sessile. This study took place at 2 sites along the Oregon coast, Strawberry Hill in Central Oregon, and Cape Blanco North in Southern Oregon. Now, both of these sites are situated along an upwelling gradient, with more intermittent upwelling in Central Oregon and more persistent upwelling in Southern Oregon. Now, what I did in this study was manipulate both of those factors I just described, light availability and herbivory. Zooming in on this photo here, you can see the black plastic mesh that was stretched over the white PVC frame there. So that mesh was meant to imitate the shading that's caused naturally by those dense phytoplankton blooms in the water. And then to control for herbivory in this experiment, you can see there's some orange paint on the rock around the perimeter of the plot. And that's copper paint. That paint excluded herbivores like limpets and chitons that don't like to move across it. So they weren't able to enter the plot. Every month, I measured 2 biological variables, kelp density, as well as the length of each individual kelp thallus within each plot. After taking those biological measurements, what I did was convert them into increments because each plot had different starting values. So for example, one plot started with, say, a density of 5, and another plot had a density of 10. So those different starting values made it difficult to compare effects among treatments over time. So what I did was standardize the metrics by taking the initial mean value at the beginning of the experiment, so month 6, for example, and subtracted that from the mean value at following time points. So I subtracted that from values at month 7 and month 8. And this way, all the plots started at 0. And then I was able to track and observe their changes through time. Now let's compare those 2 sites, Strawberry Hill and Cape Blanco North, and look at the results. I found that the effects of herbivory and light availability were pronounced at Cape Blanco North, the southern site, but negligible or non-significant at Strawberry Hill, the central site. Why did the results differ so greatly between these 2 sites? Well, variation in geophysical features, like coastal upwelling and continental shelf width between these 2 sites, provides a mechanistic explanation. So Cape Blanco North has a narrow continental shelf with more persistent coastal upwelling and strong offshore transport, while Strawberry Hill has a wide continental shelf. And here, upwelling is less persistent and currents are retentive. Now again, looking more closely at the results, let's look first at the density metric. So the figure here shows data that was collected at Cape Blanco North with month along the x-axis and density increment on the y. The red colored family indicates herbivores present. The blue colored family indicates herbivores absent. The light dot dash lines are unshaded treatments, and the dark solid lines are shaded treatments. Okay, so in the figure, what we can see is that the red colored family at the bottom there has a lower density than the blue one. And this shows that herbivores negatively affected kelp density. Now comparing the dark and light lines for each color, you can see that they don't differ greatly, right? The blues are up at the top and both of the reds are down at the bottom. So here you can see shading didn't have an effect on density. Okay, so we know that herbivores negatively affected kelp density. And now let's zoom in even closer. Because when we did that, we found that herbivores specifically reduced the survival of juvenile kelps. Okay, well, why might that be? Why would herbivores reduce the survival of juveniles specifically? Well, because juvenile kelps are tastier. They're more delicious than adults. So herbivores preferentially graze on them instead of adults, contributing to low overall population density. Now remember that previous figure for Cape Blanco North, where kelp density was greater in treatments without herbivores? Well, conversely, at that same location, when it came to kelp length, shown here, kelp length was found to be greater in treatments with herbivores. So that's an interesting difference, right? You can see that reflected in the figure here, with the red colored family having longer lengths than the blue ones. And notice that when comparing the shaded and unshaded treatments, the differences within those color families don't differ greatly. So this indicates that shading did not have an effect on length. So let's again use another lens here to analyze specifically the juvenile and the adult life history stages of these kelps, because that suggested that there were some nuanced differences when it came to light effects. Looking at the juvenile kelp figure there on the left, you can see that the longest length indicated is at the top with the solid red line. That is, juvenile kelp length was longest in the shaded herbivore present treatment. Now looking at the adult kelp figure on the right, the longest length there is indicated by the red dot dash line, which means adult kelp length was longest in the unshaded herbivore present treatment. Now, what are some possible explanations for these interactive effects of herbivory and light availability on the length of juvenile versus adult kelps? Well, let's start here at the first row and look at herbivory. So juveniles that survive grazing may benefit from grazing effects because density-dependent competition ends up being reduced. Adults benefit similarly, yet in contrast to juveniles, adults also benefit from grazing because what grazers do when they forage on kelp blades is they remove those light intercepting epibionts that are dwelling on the fronds, and this minimizes shading for adults. This allows them to grow. Now moving to light availability in the second row, juvenile kelps may be more prone to harmful effects of desiccation than adult kelps. Because juvenile kelps, they haven't developed anti-desiccation adaptations as they just aren't as well developed as those adaptations are in adults. And so the juvenile kelps in our study likely relied on shading to be able to grow in length. Whereas adult kelps can utilize light more efficiently with well-developed photoacclimation and photoprotection strategies in place against those high light levels. And so for adults, they relied on the light in our study to be able to grow an increase in length. Now, to sum up chapter 2, I asked whether the effects of herbivory and light over the population dynamics of Hedophyllum sessile could be strengthened by coastal upwelling. What I found was that more persistent coastal upwelling in Southern Oregon did strengthen the effects of herbivory and light availability on the intertidal kelp Hedophyllum. However, the direction of their control depended on demographic trait and life history stage of kelp. So for example, looking at the first box there on the far right, at the top, herbivores negatively affected the survival of juvenile kelps. Looking in the second box, herbivores positively affected the growth of both juvenile and adult kelps. Now looking at the effect of light on survival, light had no effect on the survival of kelps, yet in the last box there, you can see the effect of light on growth. Light negatively affected the growth of juveniles and positively affected the growth of adults. So after looking at all we've seen from this study, we have a better understanding of these lesser known interactive effects among light availability, herbivory, coastal upwelling, and then the macroalgal species identity and life history stages and how those all come into play. And at this point in my work, I was able to have an opportunity to scale up our experiments beyond the local and mesoscale levels and incorporate macro scale processes such as El Nino, with the 2015-16 El Nino bringing the third most severe El Nino event in recorded history. And we'll go ahead and switch interpreters.
So now in chapter 4, I ask the question, will these 3 common kelp species of the Oregon intertidal recover in different ways following the recent 2015-2016 El Nino event? And how might those patterns of recovery vary with biotic and abiotic factors? Whenever an El Nino event happens, let's just talk first about what happens during an El Nino event, which is that sea temperature goes up. El Nino events also reduce nutrients, as well as intensify wave action and raise sea level, all of which have a very destructive effect on kelp communities and reduce their populations. There are 3 major severe El Nino events in recorded history, one in 1982-83, one in 1997-98, and one in 2015-2016, shown on the figure here. And of course, we do know that climate change is happening. But how does climate change actually relate to El Nino? How might it affect El Nino? That remains unknown. El Nino does all of these things, though, in the short term, that are the same as the changes we anticipate with climate change, with climate change in the long term. So then the impacts of El Nino events on coastal environments in the short term, likely mimicking those of climate change in the long term, gives us the opportunity to use El Nino then as a proxy for possible long-term ecological responses to an increasingly variable climate. This gives us an opportunity to grab an early estimate of what might happen in the future. As you might recall from my opening chapter, I talked about how each macroalgal species allocates their energy differently among the various demographic traits. Demographic traits includes growth, reproduction, and survival, and how they allocate their energy to these different processes is different depending on the demographic traits of the species of interest, as well as their response to El Nino, and that's what I wanted to investigate. So this project, I ran this study for 3 years from 2016 to 2018 during the spring and summer season. I studied our 3 now familiar kelp species, Hedophyllum sessile, Egregia menziesii, and Postelsia palmiformis. This study spanned a total of 7 study sites over 300 kilometers of the Oregon coast, with 2 to 3 study sites within each region, northern, central, and southern. These 3 regions are situated along that same upwelling gradient I mentioned before, with northern and central Oregon experiencing more intermittent upwelling and southern Oregon having more persistent upwelling. On a monthly basis, I've measured 5 biological variables. I established 5 permanent transects for each species at each site. Transects were then sampled using the quadrat method, using a quadrat placed contiguously on both sides of the transect, and I measured percent cover, density, and maximum length within each quadrat. At each site, I also documented the growth rate for Hedophyllum and egregia, measuring that using the hole punching method. Basically what the hole punching method looks like is I punched a hole in the bottom near the meristematic region of the algae, allowed it to grow for a month, and then measured the distance between that hole and the meristematic region the following month. I also wanted to look at elemental composition. So to quantify carbon-nitrogen ratio, I randomly collected 1-inch square sections from 20 separate individuals of Hedophyllum selected at random. So now let's look at how the effects of El Nino on these 3 common intertidal kelp species might be modified depending on other environmental processes that may be happening in the environment. Let's start by focusing our attention on Hedophyllum sessile. And let's look at how Hedophyllum length and growth rate respond to El Nino. Here we're looking at the correlations between length and growth rate and various environmental variables. So what you're looking at here is called a radar plot. The little empty circles show the correlation between those metrics, in this case, length and growth rate, and the environmental variable in question. And the position of those dots shows the relationship and the strength of that relationship. So notice the dark black radial axis, that's the circle in the middle. Any little circles occurring inside that line will be negatively correlated. On the line represents no correlation, and outside the line is a positive correlation. You can see the key at the bottom there for all of my acronyms, so you know which environmental variable we're talking about. So I found that maximum length and growth rate were negatively correlated with regional temperature, as reflected in the MEI, that's the multivariate ENSO index, as well as negatively correlated with wave action given by SWHT, that's significant wave height. It was positively correlated with nutrients, the DIN, or dissolved inorganic nitrogen metric, as well as upwelling given with the beauty index. So now we're looking at the biological responses of Hedophyllum sessile over time. Hedophyllum sessile had generally a positive recovery trajectory from 2016 to 2018, with average length increasing during that time. In 2018, Southern Oregon actually had longer average lengths than other regions. So now the question arose, since Hedophyllum is a perennial algae, does that long length reflect algae growing fast, or is the length an accumulation from year to year? When we looked at daily growth rates, we found that they exhibited basically the same patterns as length among the regions. As you can see, the average growth rate represented here on this figure is fastest in Southern Oregon. So why did kelps in Southern Oregon have longer lengths and faster growth rates? Well, previous studies show that in Southern Oregon, we do have consistently higher dissolved inorganic nitrogen, or DIN, levels compared to other regions, and this may actually be providing an extra source of nutrients that help kelp grow. So what I found was that El Nino had negative effects on length and growth rate. However, these two metrics were higher in Southern Oregon, and I'm suggesting that that could be driven by higher DIN availability in the environment. So now let's look at a different metric, shifting our attention to the density of Hedophyllum sessile. Looking at density, density had different environmental controls than length and growth rate did. Density was positively influenced by waves, that's the NPGO metric. Negatively influenced by upwelling, that's the beauty index. Also negatively affected by light availability, which in that case, I used chlorophyll A as a proxy for phytoplankton abundance to calculate that, as well as negatively correlated with grazing. You might remember I mentioned the results in chapter 2. So looking at the biological response of density, Northern Oregon had a high density, whereas Central and Southern Oregon had low densities that were quite similar to each other. There was not much difference between those two regions. So why the difference? Why these differences? Northern Oregon I found that Northern Oregon had a high density primarily driven by one site. So one site within that region has a high number of recruits due to high coralline cover in that area, which facilitates the recruitment of kelps and helps them grow. The densities were low in Central and Southern Oregon. And I'm suggesting that one reason why is possibly due to high grazing activity in Central and Southern Oregon, taking that density of adults to lower levels. So what we found is that El Nino does not affect density. Density was much more strongly driven by grazing and light availability. So now let's take a look at the percent cover of Hedophyllum. What I found here was that percent cover results were basically an average of length and density results. What this could mean, I'm suggesting percent cover integrates across environmental factors and the biological responses of density and maximum length. Because percent cover reflects a species' demographic traits of growth, in this case represented by length and growth rate metrics, as well as survival, represented by density metrics, we have to be cautious since that can mask the species' internal trade-offs, which are crucial to understanding the species' responses then to their environment. So what I found was that the effects of El Nino on percent cover really just depended on the responses of the density and length metrics. So let's broaden our view and include some other kelp species here. Hedophyllum did have some apparent recovery in the years post El Nino. The species growth rate did decline, however, in 2018, as did their nitrogen tissue content. Looking now at the two other kelp species, from 2016 to 2018, their metrics generally declined as well. Egregia had low percent cover, high rachii breakage, and slow growth. Postelsia, additionally, also had low percent cover. So we just looked at each species individually and how they responded. Now let's look at the community as a whole. So zooming out to the community level, we'll look at each region. What we're looking at here is a non-metric multidimensional scaling. The position of the dots, each dot represents a kelp community for each region. Red represents Northern Oregon, green represents Central Oregon, and blue dots represent Southern Oregon. You'll notice that each dot has an arrow coming out from it, pointing to where the community changed from year to year and where it ended up. Each dot goes by month, so it's categorized by month. If you look at the figure, you'll see dots that occur more towards the top show that the community changes were more strongly correlated with the MEI, the multivariate ENSO index. Where you see them going down towards the bottom, that's actually where the community changes are more correlated with dissolved inorganic nitrogen. And where you see dots shifting to the left of the figure, community changes are more correlated with upwelling. If dots are in the same position in the figure, this means that you're looking at the same kelp community at two different time points, but from year to year in the same month, meaning if they're in the same position, they're representing communities whose composition are the same or nearly the same, whereas the further divergent, further far apart they are from each other, the more different those communities are. So looking at, I want to compare the years 2016 and 2018, highlighted in the yellow here. In Northern Oregon, kelp communities in 2018 either partially or wholly reverted back to similar abundances and composition that were seen in 2016. The same is true in Central Oregon, represented by the green dots, a very similar response with arrows moving more towards the bottom left. However, the red dots show that rather than reverting back to being similar composition in 2018 as 2016, they were more strongly influenced by coastal upwelling and dissolved inorganic nitrogen. And that was the blue dots that were more strongly influenced by coastal upwelling and dissolved inorganic nitrogen. So why did all the kelp species have such negative responses in 2016? Well, one possible explanation points to the detrimental effects of El Nino. As for the negative response in 2018, though, when we were expecting them to be more recovered, the responses could be explained by declining dissolved inorganic nitrogen levels, those DIN levels in coastal waters. You can see that levels in all regions declined in 2018. That's what this figure is showing. Basically, the responses of these three intertidal kelps post El Nino highlight the potential importance of DIN in promoting kelp's resilience to environmental changes. So to sum it up, my question was, will these three common intertidal kelp species recover differently following the recent 2015-2016 El Nino event? And how might their patterns vary with abiotic and biotic factors? Looking at all species together, what I found, looking over in that first box on the left there, the responses of intertidal kelps to the 2015-2016 El Nino did vary among species, across space, as well as through time. So focusing in on Hedophyllum sessile, what I found was that El Nino had detrimental effects on Hedophyllum. However, those effects of El Nino could be mitigated or amplified depending on the environmental processes around them, as well as kelp life history strategies. In my study, coastal upwelling provided some regional relief for kelp populations with respect to their growth needs and mitigated the adverse effects of El Nino. On the other hand, my study also showed that coastal upwelling amplified or compounded some of the detrimental effects of El Nino since it increased phytoplankton-induced shading as well as mollusk graving on those juvenile kelps, thereby reducing their density. So now we've seen various interactive effects of these processes operating on different scales to produce different responses in intertidal kelps. So now how do these processes and the interactions between them change through time? I'll switch interpreters.
All right. So we just took a look at the effects of a single El Nino phase on intertidal kelps. In chapter 5 here, we adopt a wider lens, and we're going to look at the larger relationship between El Nino Southern Oscillation, both El Nino and La Nina, and kelp abundance, and consider how their relationship might vary across sites and species. So many studies have shown that the intensity of El Nino events is often directly correlated with the magnitude of kelp population declines. For example, the more intense an El Nino event is, the larger the kelp population's decline. The kelp population declines will be, generally speaking, right? So the studies show that local processes and kelp identity play an important role in modifying the effects of El Nino on kelps. However, there have been few long-term studies comparing multiple El Nino events and their ecological consequences on coastal macroalgal communities. And the studies that are out there focus mostly on subtidal kelps. And as I said before in my introduction, kelps residing in the rocky intertidal are deserving of more attention, considering that rocky intertidal habitats do have both marine influences as well as terrestrial influences. And in those studies that did assess El Nino impacts on intertidal kelps, they did so over really limited degrees of latitude, and they were short-term, spanning usually a single El Nino event. So using this short-term information and short-term studies to assess ecological consequences can be really problematic, considering ENSO occurs over a large geographic area and may have multi-scale effects on intertidal kelps. So with this project here, I attempted to address these issues by comparing two kelp abundance data sets. One was collected during the 3 years following the 97-98 El Nino event, and then the other collected after the 2015-2016 event. And I focused on the same three kelp species, our friends, Hedophyllum sessile, Agresium zcii, and Postelsia palmiformis. Within each region, there were 1 to 3 study sites, just the same that there was with my previous project, running from north to south along the coast. Again, these 3 regions are situated along that upwelling gradient. So we see more intermittent upwelling in northern and central, and then more persistent upwelling in southern Oregon. Both of these studies used percent cover to examine changes in algal abundance across regions. Each site within these studies had 5 permanent transects established for each species, and then the transects were sampled using that quadrat method I described previously, where the quadrats are placed contiguously on both sides of that transect tape. And what I found was that El Nino effects might vary latitudinally, with negative effects of El Nino being strongest down in the lower latitudes, so in Oregon, central and southern regions. And then the strength and direction of that latitudinal trend is likely to be modified by kelp species identity and spatial variation. Looking at this figure here below, you see we have MEI, multivariate ENSO index, on the x-axis, and MEI is a metric of ENSO. You see the negative values down below on the left indicating the cool La Nina phase, and then positive values indicating the warmer El Nino phase. And percent cover is on the Y. The color code on the far right, you see Roy G. Biv backward for the Oregon coast sites, the cold colors more toward the north, and the slope figure in the top right of every panel in that upper right-hand corner there you see those thumbnails, those place the slope of each site at origin so that you can just quickly and easily compare the rate of responses that we saw among the sites. Now let's look at that last panel on the far right there. That shows the cover of Pistelcia, and it shows that it was decreased in Central and Southern Oregon as MEI increased. And it increased in Northern Oregon. Let's look at the panel to the left of that, Egregia. Now you can see Egregia responded similarly, but the panel to the left of that shows Hedophyllum, now that was an exception. The cover of this kelp decreased the most at the two far ends of that latitudinal gradient. So at Cape Blanco North at the far south end and Fogarty Creek at the far north end. Why this divergent pattern? Well, this is likely explained by some missing data in the 97-98 data set in the years after that El Nino event at both Fogarty Creek and Cape Blanco North. And so that ended up skewing the trend toward the 2015-2016 data. The next question is, well, why do we see kelps having these latitudinal responses to El Nino? I think these latitudinal responses could be explained by the weakening of the El Nino signal as it moves poleward. So El Nino originates in the Galapagos Islands and weakens and diminishes and weakens as it moves outward from that in both directions. The signal could also be further modified by variation in geophysical features, as I described before, upwelling currents, coastal geomorphology, all of that in different regions along the Oregon coast. So we just looked at the effect of El Nino on kelps, and we saw that spatial variation can modify those effects. And here we see that species identity can further modify the effects of spatial variation on intertidal kelp's responses to El Nino. So results showed that the 3 intertidal kelp species studied, Hedophyllum, Egregia, and Pistelcia, all responded variably to increasing MEI, with Egregia, interestingly, having the most negative response to the increased MEI numbers. Now we ask, well, why? Why would Egregia have the most negative response to increasing MEI across sites? This result actually is surprising because Egregia is found to be the most thermal tolerant of these 3 species. Studies have shown that Egregia may in fact be locally adapted to different thermal regimes. And if so, that actually could explain their strong negative response to increasing MEI in our study, because they were unable to acclimate to those sudden changes in their thermal environment with El Nino. But remember that previous figure showing the latitudinal responses? On the site level, Hedophyllum was the only species that responded negatively to increasing MEI across every site. The responses of Egregia and Pistelcia were mixed, and then the positive responses at northern sites and negative responses at central and southern sites that followed along that decreasing ENSO pattern. So the causes of these divergent findings are unknown and need further exploration. Now to sum up, I asked, what's the relationship between El Nino Southern Oscillation, El Nino and La Nina phases, and kelp abundance? And then how might their relationship vary across sites and species? What I found was that the magnitude of kelp responses along the Oregon coast was generally proportional to the intensity of El Nino events. Each kelp species followed a latitudinal trend, with the negative effects of El Nino being the strongest at lower latitudes, that is, in central and southern Oregon, and then decreasing northward, or poleward rather. Yet again, the strength and direction of this latitudinal trend was modified by kelp species identity and spatial variation, as we've been talking about. So bringing together the results from all of these studies, encapsulating what I've shared so far, collectively what's illustrated is that coastal upwelling is a vital oceanographic process that underpins the dynamics of intertidal macroalgae. And even more importantly, I think my work highlights the significance of three following themes in the context of coastal upwelling. First, interactive effects of key local environmental variables like light availability, herbivory, and space are crucial to understanding the ecology of kelps. So that's the first. The second is kelp species identity, their demographic traits, their life history stages, has a strong role in dictating individual, population, and community responses of kelps to their continually changing environment. And third, the combination of local and mesoscale processes and kelp species identity can modify effects of macroscale processes to produce complex, multi-scale responses of kelps across space and time. What do my findings mean for future studies? Well, the frequency of strong El Nino events are likely to increase in the future as our world continues to warm. And coastal upwelling may intensify and persist longer in that case. However, the biological effects of climate-mediated intensification of coastal upwelling on regional scales still remains inconclusive, because on the one hand, coastal upwelling could promote productivity in the region, bringing more of that nutrient-rich water to the surface. Yet on the other hand, increased greenhouse warming could cause a decoupling of upwelling from that supply of nutrient-rich water. In that case, the warming could enhance thermal stratification in the ocean, deepen the thermocline, and then subsequently reduce, or prevent rather, the cool and nutrient-rich waters from being upwelled, and instead bring up just the warm, nutrient-depleted waters. With the uncertainty that is associated with El Nino and climate change in the future, and the biological implications, the findings from this study reiterate the importance of acquiring better insight into one, how context-specific underlying conditions modify ecosystem processes, and two, understanding how each demographic trait and life history stage of kelps change with biological interactions and environmental factors over both temporal and spatial scales. Understanding these nuanced dynamics can help us anticipate future climate change ramifications and teach us how we can better prepare for them. With that, I would like to wrap up with some acknowledgements. I'd like to take a few minutes to acknowledge the many people in my life who helped me get where I am today. I have lots of things to say about each one, but for time's sake, I'll keep it brief. Of course, I have to start with my advisor, Bruce Mengi. I feel like I wouldn't have advanced in my career if not for him. His open-mindedness as he decided to take me on, right? I'm a student with a non-traditional background, and he was willing to take me on as a grad student. He saw the potential in me, and somehow he knew exactly how to turn that potential into reality. He really pushed my intellectual boundaries, and he would entice me with more research, right? Because he'd give the ecological problem at hand this air of compelling mysteriousness, which left me with no choice, of course, but to continue to discover. And at his urging, I learned to embrace the complexity of nature, to really appreciate that. And that complexity truly is the staple of Bruce's work. Brittany, Jenna, and Jonathan, when I first moved to Corvallis and joined the lab, I definitely felt like an outsider because I didn't know anyone who used American Sign Language. And Brittany, you and Jonathan and Jenna, you were all determined to learn ASL. And they did. They learned ASL so that they could communicate with me. Learning a new language in front of a native user can be pretty intimidating, but they were committed and they were open and really willing to make mistakes because they believed that I was worthy of their time and effort. And as a result of that courageous action, they made me feel like I was truly a member of the lab, and they treated me as an equal and made every effort to ensure that others saw me as one of the lab too. So they're true allies of mine. And it's an abundance of patience and kindness. And Brittany teaching me how to drive a car with a manual transmission, that skill proved extremely useful in field research. So thank you. I'm also grateful to every member of the Lubchenco-Mengi lab. And they demonstrated what it's like to have a great lab full of warm, caring people who are passionate about what they do. Everyone here on this slide showed me a different dimension of teaching, research, communication, and life. I'm grateful to my interpreters, Howleen and Deb. Navigating academia as a deaf person and the only deaf doctorate student in the university can be daunting and utterly overwhelming. And Hal and Deb are just two of the finest ASL English interpreters I've ever met. I can easily say that they truly embody what it means to be an interpreter. Their dedication to the lab's success was unparalleled. They were always eager to take on the challenge, working with me in different settings. Different is an understatement, but they'd take it on and accept the challenge. My family's been instrumental in all of my successes. They believed in me, supported me in my endeavors, and visited me wherever I was. My mom, Patricia, she is truly my biggest fan. She came to help me with my research every single year without fail. She even taught me how to solder wires together and helped me build my photosynthesis chamber. My brothers, Richard and David, they're wonderful intellectual sparring partners who always help to expand my worldview, help me think outside the box, and challenge me to articulate myself clearly. Alicia, an extremely dear friend of mine. She's the only person who truly understands the journey I embarked on. With a stroke of luck, our life trajectories have been very similar ever since we both started as two young 18-year-olds entering Rochester Institute of Technology. And because of the similarities between us, she was able to be an incredible source of unwavering support, wisdom, and love. Sanghwa was my lifeboat during my years here in Corvallis. When I first flew to Oregon for the interview with Bruce Mengi, I remembered feeling really nervous about moving to just, a whole new state, a whole new side of the country, and a small town where I didn't know anyone. During the interview, I met Sanghwa by chance. And from that moment on, we became really close friends and roommates. And every day I thank my lucky stars for the opportunity to have met her on day one of my journey. Mandy's an adventurer, and when it came to navigating through my tumultuous journey as a doctoral student, she was by my side. She consoled me when I struggled and cheered me on when I made strides. And then she was willing to sit back through countless hours of watching me practice my presentations and giving really valuable feedback. She was always ready to quench my urge for adventures as well. Nathan made me believe that I was actually worthy of the great Bruce Mengi. He helped me see my perceived deficiencies as a source of strength and encouraged me to take a big step outside my comfort zone. Miriam, she showed me what it means to grab life by the horns and embrace the unexpected. She introduced me to the untapped beauty here in Oregon. And she also shared in my excitement for outdoor adventures. So we shared that together. Camille, a deaf marine biologist from France, she showed me true joys of working with another deaf scientist and communicating in several languages while doing science. So truly grateful for her. Toby has been an unwavering beacon of light during this final part of the journey. He helped me find my momentum during the final stretch when I felt like there was no end in sight and sometimes no light at the end of the tunnel. He really helped push me through. I would like to also acknowledge the deaf community, especially Rochester's deaf community, that raised me and gave me the beauty of American Sign Language and deaf culture. Taught me to be proud of my deaf identity and always supported me in all my endeavors from near and far. And I also thank other interpreters and countless volunteers who helped me in the past with both communication and research. And then I want to thank my graduate committee, Mike, Patrick, Tiffany, Sally, for their guidance on my research. OSU's disability access services for being open to the paradigm shift of what it means to provide accessibility and supplying high quality interpreters for all aspects of our program. And the OSU Integrative Biology Department, especially the 4 Ts, Tara, Teresa, Tracy, and Trudy. They patiently helped me navigate through the complexity of a graduate program. And finally, my research would not have been possible without the funders listed here. I'm eternally grateful to each and every one of you. And one day, I hope I can do for you what all of you have done for me. Okay, at this point, I'll ask the interpreters to go ahead and turn on their videos. And then I'm going to stop screen sharing. Okay, and at this point, if we're ready, I'm going to go ahead and start the Q&A section of the talk. And due to time constraints, I'm only going to be able to answer a few questions, but we'll get that started. And I'm going to give Teresa a little bit of time to see what questions are out there and for me to peruse myself here. And Deb's saying, I'm not sure that everyone can see you, Barbara. And Barbara says, yeah, I'm not sure why that might be. Teresa, could you, on your end, select gallery view? That might be helpful for participants to see. Okay, can everyone see me now? It looks like everyone can. Okay, great. So the first question, is there any particular reason why you chose those 3 species of algae to focus on for your research? Well, those algae are very common on the Oregon coast, and there was a great abundance of data on them as well, so I was able to look more closely at the whole host of things that I looked at, right, with respect to them. They're really important to the intertidal community. They provide habitat for different organisms, as I explained in my talk, et cetera. So that was why I chose those. Second question I'd like to answer, let's see. Growth rate and length has come up frequently in your talk. Can you please talk about what the significance of length is when it comes to kelps? Sure. So growth rate and length both generally are correlated. And so if something grows more quickly, in general, it's going to be longer as well. But sometimes you can glean a little bit different information from both of those measurements. If something grows really quickly, but doesn't grow very long or very tall or very large, that can happen because there's a lot of wave action that can actually kind of destroy or take away from its length. Growth rate gives you a more immediate picture of kind of what's going on for the record. And then length overall gives you a bigger picture over a longer span of time about kind of what's going on in that algae. Okay, next question. Juvenile kelps, why are they tastier? Well, this could have to do with the plant's secondary metabolites, or they're asking if it could have to do with the plant's secondary metabolites. Yeah, the specific chemical makeup of the algae, which I'm not particularly familiar with, but algae and kelps in general, they're more plastic and just tender when they're young. And it's the same with other animals that, you know, like we eat veal. Well, why? Because yeah, they are tastier. They're more tender at a younger age. And as they get older, because as things get older, they establish chemical defenses and they get tougher. And so for herbivores in particular, they aren't as tasty. They don't like to eat them at that stage. Okay, moving on to the third question here. Let me take a look. Okay, this question says that the graphs showed that growth in 2016 and 2017 increased, but when it came to 2017 to 2018 in particular, growth was reduced. It took a nosedive. Why was that? Well, when El Nino occurred, kelps were decimated and then needed to recover. In the year 2018, yeah, there were lots of metrics that were decreased, and we think that it's because something else dropped in the environment, particularly nutrients. And we don't know what drove that, what the particular factors were there. We definitely need to look further into that, but there are other things in play. It also showed that kelp responses are tied to nutrients in the water, and that when nutrients increase, it does promote growth for kelps.
And we do know that dissolved inorganic nitrogen did go down. So that could be part of it.
Okay, so I'm just going to answer just a couple others, and then I'm going to go ahead and wrap up, and I'll wait for a few more questions to come in. Okay, so now we'll go ahead and switch into Word.
And yeah, anyone who wants to ask a question in ASL, go ahead and raise your digital hand, and that'll let Teresa know, and she can promote you to panelist so that we can see you on screen if you'd like to ask your question in sign language. Let's see, watching. Okay, so while we're waiting actually for somebody to come over, nutrient increases seem to have a positive impact on kelp, especially in the context of climate change. If the ocean is feeding those extra nutrients to the kelp, will that kind of help combat some of the negative effects of climate change? Well, that's a great question. And what my research shows is that it does seem like nutrients do help kelp get by in those times where, maybe there's other stress on them. But I want to add too that, it can kind of send the ecosystem kind of spinning for a loop a little bit. When you've got an invasive species, for example, as a, you put an invasive species in, and now there's a predator there that's never been there before, or there's a grazer there that's never been there before. And just having that new invasive species in the ecology has a big impact, and it can have cascading effects on other things, right? So sometimes it's a viable option, and other times we have to look at, sort of the consequences of that as well. So in that similar vein, I would say hard to predict.
Hi, Barbara, it's good to see you.
Yes, good to see you, Caroline.
I want to say congratulations on your defense. So my question is, you had mentioned how juvenile kelps are more attractive to grazers as opposed to adult kelps. What's the difference in nutrient value between adults and juveniles? I'm just interested, what makes juveniles more attractive to grazers?
Yeah, that's a good question. As far as different nutrient value goes, there could be a bunch of things going on there, and I don't actually know. The most common reasons given is that as adults grow bigger, they have a different phytological makeup. So basically, they're developing chemical defenses as they get older and older to fend off grazers particularly, and those chemical defenses make them not taste as good, and so grazers prefer not to graze on those. So juveniles haven't had a chance to develop those yet, and thus grazers just are not as interested in them.
So it's not due to the carbon-nitrogen ratio of the kelp at all. That doesn't have an effect. I mean, because I was thinking, what's greater? What has a greater effect? The effect of nutrient value or the effect of the palatability that you were describing?
Kelp physiology is not my area of expertise, actually. But definitely, I would assume if you did an assessment on carbon-nitrogen ratio, you might find that palatability is in fact affected by those ratios. I can't address that specifically. I don't know.
Thank you.
Okay, let's take one more question, and then I'll go ahead and wrap up. So this question is, with regard to climate change, kelp could possibly adapt, and would they maybe evolve? Would we see some evolution through time? That's a very broad and great question. Certainly we can look at history to know that most things do evolve over long enough periods of time. The problem is if we're looking at specifically how kelp adapt or evolve within the space of, within what's going on with climate change, within that context, we actually don't have a long enough period of time where we've been seeing climate change. That's only been in the last few years. Most evolutionary changes that we know of throughout history take place on the scale of hundreds, 400, 500 years before we can start to see those things. So I would say we might be able to look at DNA, like variations in DNA change or genetic changes, but to be able to assess those genetic variations over time. And certainly if we see the temperature keep warming, then that would be something that we would want to maybe investigate further, seeing which of those algae might be the most thermally tolerant, and which of the kelps, develop sort of survival strategies in the new warming climate and which ones disappear. That'll be a really interesting thing to investigate as time goes on. Okay, I think I'll go ahead and wrap up. Thank you so much, everybody who came. Thank you for coming to hear my talk, and have a great day. Bye.
Okay, the committee, the next meeting is with the committee, and it's about 26 after 11. So let's meet, let's say about 25 to 12. And I've sent you out the link. If you don't remember it, don't have it, send me an email and I'll forward it again. And Barbara, thank you for a fantastic seminar. That was just great. So we'll see you shortly on the other Zoom. All right.
Awesome. See you soon.
Thanks. Bye.